Sir? Sir? Sir, suddenly there is a...
Well, luckily, I'm in a place like which I feel like it's an oyster. The support of the management, the support of my fellow teammates, the organizers, organizing committee, my faculty members, support of the students, and with all these, supports, I am able to do what I am doing now. Coming back to this program, I'm happy to share that almost to the tune of 570 registrations have come to this particular program. It's far better, than the session 1 of this impact lecture series, which was only up to 400. This thing, online programs are being conducted by several institutions. I advise all the delegates of this program, and also in this program we have several stakeholders like principals are there, research scholars are there, students are there, everyone. I advise everyone to attend to such programs, as many as possible, which definitely enriches our knowledge. Knowledge is not simply reading or listening. Definitely it is also depends upon the dissemination also. Okay, only when we keep on learning, keep on listening, the things we get to know more and more deeper knowledge. Session 1 on July 25th, June 25th we have conducted, and it included 2 great resource persons. One is Mr. Alluri Krishnaraju, head of the project management, pharmacology and clinical research, Lila Nutra R&D, Vijayawada. The second person lecture was by Professor Ragala Ravikant. Is a startup mentor, entrepreneurship motivational trainer from Hyderabad. Today we have 2 eminent people again, who is, you can see, a startup owner, Mr. Suresh Kandagatla. Today morning we have a session by Mr. Suresh Kandagatla. He is going to deliver a lecture on pharma startup, an incubator for innovation. Second one, in the afternoon session, we have one Miss Nisha Wadhwa. She is an associate vice president of Aumira Intellectual Properties, New Delhi. All these topics are very aptly coined, to enrich the knowledge of the students, not only the students, but also the like faculty members. If the student listens some knowledge, gains some knowledge, it will be restrained to him only. But if a teacher gets the same knowledge, it will help to light so many lamps of, to help increase the knowledge of many other of his or her own students. Today's 2 topics: intellectual property, the patents, turning the patents into profit-making assets. Patents, word nowadays. Every organization, like every college, institution, they're all going for accreditations. Accreditations are the ones which shows the transparency that is happening in the institution or a hospital. It can be any, it can be a hospital, it can be an industry. You all must be knowing FDA approvals, GMP, and all these things. For institution, academic institutions, it is like starting from ISO, NBA, NAC, and all. These all accreditations, even for hospitals.
Sir?
And extend their accreditation to our institutions. I'm happy to say that we are on the way. That field. Taking this opportunity, I thank the management, Narasaravapet Institute of Pharmaceutical Sciences, for extending their support and also to organize such several programs in the college. And also the MHRD's Innovation Cell, the team of to our college, the team members led by Ms. V. Swati and Mr. M. Ramakrishna, Ms. M. Ramiteja, and Mr. Mullah Mubin, the coordinators, and from various facets, the registered delegates especially. I extend my sincere thanks to all, and I'm sure that you'll be having next half an hour, next 1.5 hours, a session by Mr. Suresh Kandagatla, and in the afternoon by Mr. Vadva. Thank you very much. Thanks for giving an opportunity. Have a nice day. Thank you, madam. Over to the technical team.
Thank you, sir, for your brief introduction on the programs that are being organized previously as a part of the phase 2 session 1, and today as the session 2. So now I invite Mr. Chukka Ajay Kumar, Assistant Professor, Department of Pharmaceutical Chemistry, to brief out of the today's resource person, Mr. Suresh Kandagatla.
Good morning, myself Chukka Ajay Kumar, Assistant Professor in Narasaravapet Institute of Pharmaceutical Science, Pharmaceutical Chemistry Department. I am pleased to invite Mr. K. Kandagatla Suresh to the gathering. Sir pursued M.Pharm in Pharmaceutical Chemistry, JNTU Anantapur, and B.Pharmacy in JNTU Anantapur. Currently, Sir is the CEO of Biogenic Products Private Limited. These biogenic products private limited is the natural products based research and development company working under the Osmania University technically business from the year 2020. Sir previously worked as an assistant professor in Vijay College of Pharmacy and St. Mary's Pharmacy College. Sir presented various guest lectures in various colleges and coaching institutions. Sir has qualified GPAT and NIPER examinations. Sir has published around 8 research papers in national and international journals. Looking forward to wonderful session, sir. I thank everyone for providing me this wonderful opportunity.
Sir Suresh, sir.
Good morning to everyone.
Good morning, Dr. Sir, Mr. Suresh.
Good morning, sir.
Happy to see you at our college. Thanks for immediately readily accepting the request. And definitely, I'm sure that your lecture is going to be a very beautiful one for all the audience, especially the students who are sitting in the seminar hall. Thank you and welcome you to NIPS.
Thank you, sir. Thank you, Principal Sir. Actually, my humble apologies for the entire team and participants. So I have been late by 30 minutes because of meeting with VC.
No worries, no worries. We are almost in time. Don't worry.
Okay, sir, thank you. Sir, can I present my PPT?
Madam, is it okay?
Yes, sir. Already I have given you as co-host, sir. You can share your screen, sir.
Okay.
Thank you. Please.
Okay, let me share my PPT. Hello.
Yes, yes, you're audible.
Recording in progress.
Sir, you can share your screen, sir. Participants, delegates, please be online. Just I'll check the technical error. Please be online. Sir, Suresh sir, I'm sharing the screen. Just please check, sir. Suresh sir.
Please call sir once personally, madam.
Yes, sir.
Sir, voice రావట్లేదు. Sir.
Sir, please kindly unmute because I have already given you as co-host, sir.
Hello.
Sir,
Ma'am.
Suresh sir. Mr. Suresh, come here.
I'm audible now.
Sir is audible, but lag is there.
Okay. Yeah. Am I audible? Hello?
Sir, no, sir, there is disturbance, sir.
Is audible? What's the problem? Ma'am.
Hello. 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 Hello.
Hello, all the delegates are requested to unmute your, to mute.
Hello, Suresh Garu.
Yes, ma'am.
My sincere request to all the delegates, please switch off your mics. Please mute them. So because of some technical errors, Sir will be online. Sir is facing some network issues. Hope you understand.
Madam, is it audible now?
Yes, sir. Yes, sir. It is audible, sir.
Okay. First, thank you, Narsarapet Institute of Pharmaceutical Sciences, for giving me this opportunity. So, today I'm giving my experience with incubators, startup incubators. So, how the startup incubators is.
Hello.
Sir,
Startup
Suresh, sir,
incubators,
please
how.
carry on.
Okay, startup incubators. So my company name is Biogenic Products Private Limited. Madam, next slide, please. So we are a group of 3 people. We have started this company in January 2022. And before that, I want to tell you how this journey began in the startup. So how this idea came and how this idea entered, converted into a startup company. So we are Indians and we are from village background. We 3 are from village background. And what I want to tell you is, whenever we used to get any sickness, we use home remedies or herbal medicine. Generally, 80% of Indians use the herbal medicine. So
Hello. Hello.
Madam, I'm getting some disturbance.
Sir, I am also unable to identify the person, sir. I am trying. I will check,
Okay.
sir.
Okay, no problem. So this is it. So as we use home remedies and herbal medicine, so herbal medicines. Herbal medicine. So these herbal medicines are posed to so many issues. So later in the session, I will discuss what are the major things. So we have started this company in a vision to become the leader in this healthcare sector, especially natural product related healthcare sector. Madam, keep the previous slide. Natural products related healthcare sector, especially we are targeting obesity, and our mission is to discover the standardized natural products and natural formulations. So this is what it. So we are at the group of 3, and our founder is Dr. E. Sujata. He is a professor in the Department of Botany, Usmania University, and she is the spearhead of this company. And myself, K. Suresh, and I have completed my doctors in pharmaceutical chemistry recently. And Mr. Sudhir Kumar, another one partner in this company, he is a natural products expert, and he's also completed his PhD from NIPER. So, madam, next slide. So first thing, any idea. So how any idea always generates from a problem. So when you are entering into the startup business or any other thing, if you want to grow a business, if you want to start a business, there must be you must solve one thing. You must solve something that is a
Hello?
problem.
Hello.
Something that is a okay. So the problem we have identified. So how the idea we got. So this is the problem. So first, this is a problem. The idea we got a problem is once we have we are doing our regular research in our college in our college, and we are publishing the paper. We have studied that. So herbal extracts or herbal medicines, they are in the long-term utilization, they are prone to some health issues in elder patients. So most of the herbal formulations taking or herbal formulation taking persons, they are suffering from kidney failures and liver damages. So we have made some small literature review and small research with anyone wants to speak?
Yes, sir. Yes, sir. You can continue, sir.
Okay. So first thing, identification of the problem. So the problem we have identified is two things. Two problems we have identified regarding the herbal materials. So first thing, the herbal extracts. The raw herbal extracts and nutraceutical products, they suffer low content of active principle. Low content of active principle. And second thing, higher amounts of unnecessary toxic chemicals. And third thing, needs a longer term administration. And the second major thing we identified is adulteration. Adulteration of the herbal medicine. So there are two ways of adulteration of the herbal medicine. So that is substandard raw materials utilization, and second one is synthetic chemicals. So now I will explain the problem one by one. So first thing, regarding raw herbal extracts and nutraceutical products, low content of active principle. So what is this low content of active principle? So let me tell you about the example regarding this one. So there is one company in India, it is preparing ashwagandha extract.
Hello?
Extract. I think everybody knows the ashwagandha about ashwagandha. So students, what is the use of ashwagandha? Anyone? Can anybody tells me the use of ashwagandha? As we are a pharmacy students, we know about it. Okay. Let me tell you that one also. So it is a immune booster. is an immune booster. So generally in any herbal medicine, the ashwagandha content should be more than 10%. Should be more than 10%. That means ashwagandha contains vithanolides. It should contain vithanolides more than 10% in the extract. That means only the 10% is the active principle, but the remaining 90% is vegetative matter. Remaining 90% is the vegetative matter or other constituent present in the ashwagandha extract. So now this company we are talking, they are telling that it is a 10% ashwagandha extract and it will give you better immunity boosting capacity. But in the original scenario, it contains only 6% ashwagandha. That means 6% vithanolides it contains. So only the 10% vithanolide content ashwagandha is helpful to boost the immunity of our body, but the 6% cannot help. So this is the problem. So they don't know. Most companies they are giving, they are doing this thing. So they are not going for the original content. So the pharmacopoeia and pharmacopoeia they have taught us this 10% extract will be very good, not 5%, 6%.
LAC.
Problem. Low content of active principle. So next one, higher amounts of unnecessary materials, and I have covered this also. So higher amounts of unnecessary toxic chemicals means other than active principle. Suppose this 10% ashwagandha, there is 90% of the other chemicals. 90% of the other chemicals. So these 90% maybe may not be useful to our body, but that's why unnecessary or sometimes they may lead to toxicity in our body. So that's why they are toxic chemicals. And another major problem suffering from this herbal medicine is you have to administer for a longer term. So generally herbal medicines, whatever the herbal medicine you need to utilize it for months, then only you will get the better effect. So second major one, adulteration of the herbal medicine. So the herbal medicines or the raw herbs that are utilized for the herbal medicine will be substituted with other raw herbs and synthetic chemicals for the better effect. So next slide, madam. So one of the best example for this thing is Chyawanprash. So you know Chyawanprash is utilized for the immune booster and strengthening of the brain. All these things are the major uses of the Chyawanprash. But the original Chyawanprash contains the 8 ingredients. So the Crepidium acuminatum, all these 8 ingredients are the original things. Ingredients in the Chyawanprash, but in India now they are replacing these 7 with 4 substituted or adulterated adulterated plant materials. So these are Ciconia digitata, Dioscorea bulbifera, Withania somnifera, Aspergillus decimalus. They are adulterating these things with this one. So now next slide. So now the solution we have given is active fractions. So active fractions are, let me say in this way, generally we will do the extraction, and that extract we will directly give it in the form of tablets or hair capsules or any other formulation. But the active fractions are purified. Active fractions are purified mixtures of the phytonutrients. That are available from different separation techniques. So here in this separation process, what we are doing is we are making the extract so sure that it doesn't contain any harmful chemicals or any unnecessary chemicals or any unnecessary phytochemicals. Strictly speaking, phytochemicals. So instead of, suppose if you have taken an extract, that extract may contain 20 types of the phytochemicals, but in the 20 types of the phytochemicals, the original activity, the bulk activity is due to either 1 or 2 or 3 of the phytochemicals, but not all the things. So we are avoiding those things. We are separating the active constituents from the non-active constituents. This is our technology. So this is the center point, and this is the point where we have based our company. So the primary objective of our company is to create this active fraction or utilize innovative methods or separation techniques to help to create these active fractions that contains only active principles, 1 or more active principles, 2 or 3. So whatever it is, they will have only the active constant that is responsible for the biological action. So thereby, what is going to happen? First thing, we are removing the non-active constituents. So along with non-active constituents, we also remove the toxic chemicals. That's why these are toxic. These contains no toxic phytoconstituents and reduction. Next thing. So after doing this, what is going to happen? So once we have done this, initially we need to utilize more amount or higher amounts of the doses, because along with the active constant, it contains some other unnecessary chemicals. We need to take a more and more amount of the drug. So now our extract. Now we are only taking the active fraction. Active fractions are very less, and they are the sole responsible for the
Hello.
biological activity. That's why
Hello,
we
Wazir.
can reduce the
Wazir.
reduction in the dosing frequency. So this leads to the reduction in the dosing frequency.
Wazir.
Next thing.
Hello.
Okay. Next. So if you reduce the dosage, what is going to happen? Simple. It will increase the patient compliance, because instead of taking 1,000 mg herbal medicine, going to take only 100 mg herbal medicine. So what is going to happen? The bulkiness of the drug, the bulkiness of the medicine is going to decrease, and we are reducing the toxicity because of the purification process. And now this leads to high compliance with the end users. So because anyway you are reducing this thing and you are increasing the potency. So furthermore, we have added some other thing into our product. This is called value addition. This is called value addition to the product, that is nano formulations. So everybody knows that nano formulations will increase the potency of the compounds, That's why once you have prepared these active fractions, why can't we prepare a nano formulation from these things, so that we can further reduce the dosing and further increase the potency, So this leads towards the
Sir, Suresh, sir. Sir, your voice is not audible suddenly.
Is it audible now?
Yes, sir. Now it is audible.
Okay, can you share it again?
Sir, already screen is visible, sir.
But it is not visible to me, madam.
Yes, I'll check again, sir. Just a second. Sir, is this visible now, sir? I request any of the participant also please respond. Is the PPT visible or not?
Yes, ma'am, it's visible.
Okay, ma'am. Thank you so much. Suresh, sir.
It is can you present it, ma'am?
Sir, is it visible, sir?
Yes.
Okay, sir.
Present it, madam. Okay, thank you. So nano formulations, this is called value addition in terms of business terms, value addition. So value addition means increase its intrinsic value. Increase its intrinsic value. Already the purified fractions are having a value in the market. So, but how can you get more from that one? How can you get more from that one? So by includes one technology. So here we have added one new technology, it is nano formulations, nanotechnology. By utilization of nanotechnology, we are increasing its potency and we are increasing its value. So all these things. So how can you achieve this thing? So only with high quality materials, only with high quality materials. That's why we make sure that we are in continuous contact with the farmers. So we have prepared 4 different products. So for those 4 different products, we are the cultivators. We are cultivating the materials. That means raw materials, raw herbal materials, and we are making the entire management of the materials. So that's why we can assure the quality. So this is also a ultimate selling proposition. So here in the solution case, I have put one bracket here, that is USP. USP means unique selling proposition.
Please switch off your mics.
Shall I continue?
Yes, sir. Yes, sir.
Okay. So we have made it. So we want to give the quality material or quality end product. So we need to keep the supply chain in our hands. So that's why we have created a group of farmers, those who will farm their lands with the raw materials, whatever we require. So we have made this pact with some farmers around Rangareddy district and Nalgonda district. So we have made this thing. Now, next. So this is the thing. So if you are really interested in this lecture, so listen to me, listen to this slide. So whatever the thing that is we have done. So now I'm explaining so how it is going to happen with the industry or with startup ecosystem. So how the startup ecosystem works. So first thing, if you are having an idea, so discuss with your friends, discuss with the teachers, discuss with your professors who are there. So first thing, we will do that one. So any startup business will flow in this manner. So the first thing, idea generation. The first thing is idea generation, and second thing is idea screening. So once it has done, idea screening is done, then we'll think the proof of concept, whether this idea is having any scientific base. You got an idea, it is having any scientific base. So we will develop that concept and test that concept. Then we will think about the marketing strategy. So once the proof of concept is done, So once the proof of concept is done, that so what is the market strategy that we can sell this product to the people or to the some other company or any other buyer, willing buyers. So how can we do this? Then we'll go for the product development. So with this product only we can sell those things. But initially we need to see the market survey, because whether you are having an idea, but that idea may be in the market already. That idea may be in the market already. So we need to check whether the market is saturated with this idea or not. So if it is saturated, then what is our uniqueness? So why the customers will choose our company products rather than their company products, the existing market company products. So how can we enter into the markets? So that is market strategy and business analysis. Next, the thing is product development. So we will develop the product based on the market needs and the remaining things. Then we will deployment. The deployment means now we are testing our product with the market. So once the market accepts or rejects depends in this stage. So once it is rejected, then we will again go back to the ideation stage and we will start it again. So this is the thing. So once the deployment has done perfectly, that means we are successful in the market testing. Then we will go for the bigger market entry. Bigger market entry, that is the manufacturing scale. So up to deployment, it is in the R&D scale. After that, it is in the manufacturing scale. Here from R&D scale to manufacturing scale, we need to increase the potentiality of our production levels. So now I have explained till now idea generation and concept development. So now market strategy, remaining things we will see. So in the case of idea generation, I told you how we got the idea. So as because there contains a more number of toxic chemicals and less number of active constituents and adulteration, we have find the problem. So this is the idea generation. Next, idea screening. So whether this idea is correct or not, whether it is having a validated base or not. So for this one, I have explained that. So already in the market, they are doing something, but it is not up to the market. So we can, this idea is a validated idea based on that thing. So next, concept. So here the concept means, so how you are solving the problem. So we have, I have told you that the problem we are solving through active fractions, So this is the concept. So we have developed this concept. So active fractions are useful and active fractions are going to give us, and testing of the active fractions, whether the active fractions will be sustained in our market. So we move to the market strategy. So once we have entered into the market strategy, so we know that herbal medicine market, listen carefully, herbal medicine market is already saturated. Number of companies are there in India. Around 6,000 herbal medicine companies are there. Herbal medicines and nutraceutical companies are there, but still we want to enter. So already saturated market, how you are entering and how you become successful, that is a very biggest challenge for any company. So, but the advantage we are having, the unique advantage we are having is we are not giving the herbal medicine in the what the 6,000 companies are giving. We are giving them in the purified fractions. We are giving the only active fractions, not the extracts, what the companies till now are doing. So this is the unique pivotal point that is differentiating our company from the rest of the 6,000 companies. And on this basis only we have incubated at Osmania TBI, Technology Business Incubator. So with this only one point, active fractions only, we have convinced the entire team of the Osmania TBI, Technology Business Incubator. So once we have presented this idea, so they have validated. After the validation, they told that, so market strategy. So what is your market potential? Then only you can survive. So they, after due diligence from those TBI for Technology Business Incubator analysts, so they come to us and we have explained these things, and so after that only they have applied, or they are they have accepted our invitation to enter into the R&D activities. Kindly mute your audio. Okay, thank you. So this is the thing. So how we agree, or we made them to agree on our terms. So once our presentation done and everything done, they have given the space now. So till now they haven't given any space in the Osmania University to work on our idea. So after that, after this active action based, and after we have shown the market opportunity for this type of the products, because it is already saturated market. This is the first question we got from the investors. So it is already saturated market. So how you are going to survive with this, sir? It is a saturated market for the regular herbal medicine. This is not a regular herbal medicine. We are preparing the herbal medicines in the active actions, and that active actions we are giving them into the formulations, nano formulations. So no herbal nano formulations are available till now based on active actions. So this is the unique selling point, or this is the unique selling proposition for our company, and we will prove this concept. So after that, they have agreed to this thing, and they have given the lab space to us, and we have started the R&D. We are successful in R&D scale now. Now we are moving into the manufacturing scale. So once this was done, so product development. So we have developed 4 of the products, and in these 4 of the products, one of the product already entered into the market. So now we are in the phase of market testing. So once that market testing is done, we will go for the deployment, and after that we will go for the manufacturing scale. So till now we are in the R&D. We have successfully completed our R&D testing. So now we are moving to the manufacturing scale. So now the thing. So how technology business incubators, how the Indian government is helping the startups. Indian government is helping the startups. That is a very, very important thing. How a small idea from our brains is incubated in the TBI, and now it was taken up by the TBI into a next level. So this is the very, very important thing. Madam, please keep that thing. Last one, this one. So listen carefully here. So once we came here, we came with a small idea. We came with a small idea, and in the startup ecosystem, the technology business incubators, these are started by our Indian government. These are started by our Indian government to commercialize the ideas in different sectors. So here in the Osmania, we are also having an incubation center. This incubation center is helping us now. Till now, it helped us so that we can, from R&D to, we are going for the manufacturing facility now. We are increasing our capacity to manufacture the products. So till now we have prepared the products, and we have innovated the products, and the remaining things taken care of by the technology business incubator. So come with any idea, whether it is a small idea or a big idea, or it is a critical idea, or it is a non-commercial idea also. Business incubation centers will think on it, will have a brainstorming sessions on that, and they will come up with a solution for your problem. And now they will give your idea to some other investors. Investors means those who are having money and willing to invest in your idea and see whether it is working or not. So this is the very biggest and very good thing about the technology business incubators, because they make sure that your idea will be successful. So that's why incubation centers are very, very, very important in the startup ecosystem, because they know the links. So if you are struggling with any problem, suppose any problem, they will attach you that persons who can solve that problem, who can solve that problem. So can you go for the next slide, madam? So stages of innovation. This is our company's innovation stage. So how we have one step by step we got this thing. So first ideation stage. So ideation stage means idea. So we got one idea. So what is that idea? We want to create an active fraction based formulation. So okay, you will prepare that active fraction based compounds, but which area you are targeting? That means which patient area you are targeting. We are targeting the lifestyle disorders. We are targeting the lifestyle disorders and proof of concept. And now coming to proof of concept. So the proof of concept is simply literature. Simply the literature. So previously, how many members have done this type of work and whether they are successful or not? Whether any research and development activities went on this manner. So we are having a number of literature support for this thing, and we have shown the proof of concept to these things. Next slide, madam. So innovation. So how our innovation stages of innovation. So how we have conducted our experiments and R&D success happened. So this first thing. So as we are targeting a natural products, so major source is plants. So first plant sources. Then after that, so we have done what we have done is bioassay guided fractionation. Bioassay guided fractionation means very simple. So I think in your B.Pharm final year or third year in the pharmacognosy, you are having extraction with the Soxhlet, maceration, percolation, different techniques, supercritical fluid extraction, everything is there. So we'll do the by selecting different solvents like non-polar, hexane, ethyl acetate, methanol, chloroform, methanol, ethanol, water. So one by one, we will repeatedly extract the same material with different solvents, and we will check the activity. We will check the activity for the compounds. So whatever the extract, suppose if it is active, so for example, take anti-diabetic activity, or here in this case we have taken the obesity. So here in this case obesity. So those fractions, suppose hexane or methanol fractions are given anti-obesity effect, then we will take only those two fractions and the remaining extracts we will discard them because they don't show any activity, but these two, hexane and methanol, have given the best activity. So take these two extracts. Now purify these two extracts. Purify these two extracts into active fractions. So once this process has done, what we have done is we have taken two approaches. One is isolation of the compounds, isolation and characterization of the compound, and second one is formulations, advanced formulations with nanotechnology. So this is the area we are marketing now. So after purification of active fractions, the first one is isolation and characterization. That is the biggest sector, very big sector, and it needs a huge amounts of money. So we are not entering into that market, but we are entering into this market where we can easily prepare the formulation. So these active nano formulations will be patented, and we have given this to the preclinical studies. Now we are in the stage of preclinical studies, where two preclinical studies are completed, two products preclinical studies are completed, and the two products are going on. So after clinical trials, we will go for the patenting and manufacturing. So this is the stage. So these are the stages of our company working style. So from plant source to finally advanced formulations, we will prepare. Those advanced formulations will be tested in vitro and in vivo activities. Once in vitro and in vivo completed, we will go for the clinical studies. After that, we will go for the manufacturing. So now we are at the stage of preclinical studies, and one of our product, one company has taken up the clinical trials. So because we have a MOU with them, and they are taken up our product, they will brand it on their own name. This is called white labeling. So product is yours, but you can't sell it on your brand. So this is called white labeling because as a thing, we need to sustain. So in order to sustain, we need to generate money. So for initial generation of the money, we are approaching companies who are in need of the formulations. We are preparing the formulations, we are giving them the formulations, and we are getting the licensing fees back as a royalty. So this is what happening. So next slide, madam. Next slide. Okay, working methodology. Working methodology for prototype. Prototype is nothing but your product. First form of your product is prototype. So how can we do? So first we went for the collection of the material. So here we have collected. How we have collected? We went to the farmers. We have given them the seeds and propagation methods, and you give us the final end raw material after harvesting. So we have collected. That means we have harvested those things and we have collected. And before that one also, after that one also, we have authenticated these materials with the botanists. From Botanical Survey of India, we went to Botanical Survey of India. We approached them for the authentication. So because even though if we are thinking that it is a perfect raw material, perfect plant material, but it is not, it may not be. So that's why for authentication we have done this. Next, after that, drying process. So the drying only under shade. Only under shade drying. So only under shade drying we have to do. Why? Because raw materials contains so many phytochemicals that are heat sensitive, that are sensitive to sunlight. So here we only go for the natural drying. Natural drying, that is under only sunlight, not shade drying. So next thing, pulverization. Pulverization is nothing but simply reducing the size of the product. So we take the seeds, so we'll convert it into a powder. This process is called size reduction. This is called size reduction. This is called size reduction. So next thing, the process contains extraction and with different extraction techniques. So generally in the bioassociated fractionation, we utilize hexane, ethyl acetate, methanol, and water, or mixture of any of these things. So after that, extraction techniques. So generally we utilize the Soxhlet extraction as it is a very useful and most efficient. So next slide, please. So this is called next biosegregated fractionation. So once we have done with extractions with the different solvents, so each fraction, hexane extract, ethyl acetate extract, and methanol extract, water extract. So we will have a different extracts of the same material. So we will take the hexane extract, ethanol, methanol, water, ethyl acetate, and for each one we will do the in vivo bioassay and in vitro assays. In vitro and in vivo assays. So once we have done this thing, we will know so which extract is having activity, which extract is having the activity. So we know that. So once the extract is having activity, we will prepare them into isolation. So for the active fractions. So even in the hexane also, suppose take that in the hexane extract, it has given the anti-obesity activity. Both in vitro and in vivo, it has given the activity. So now it is confirmed that only hexane part is confirmed. That means the components that are the phytochemicals that are dissolved in the hexane having the property of to reduce the obesity. Having the property of reducing the obesity. Now we know that hexane extract is having the activity. So now in that we find from PLC or some other analytical technique, it contains a 22 compounds. It contains 22 compounds are there in the hexane extract. So in the 22 compounds, which compounds are giving the activity? compounds are giving the activity? We don't know. So now what we will do, we will make them, or we will separate them by utilizing the chemical separation techniques, into different active fractions or different fractions. Now, hexane extract contains 22 chemicals, but in the 22 chemicals, how many phytochemicals are responsible for the anti-obesity action? We don't know. But now what we know is we can separate some of the phytoconstituents. So now initially what we have done, madam, keep the last slide. Initially what we have done, so isolation, how can we isolate it? We utilize two techniques, column chromatography and chemical separation techniques. So by column chromatography and chemical separation techniques, we have separated those 22 compounds into a six fractions. That means one fraction contains two compounds, one fraction contains four compounds, and another one fraction contains only one compound. Like this, we have separated the things. So these are now called as fractions. So now we are selecting. Again, we are doing the in vitro and in vivo assay for these things. So once we have done this, we know which fraction is responsible for the action. So once we know that which fraction is responsible for the action, then we are making it into a formulations, nano formulations. So next slide, please. So this is the way. Next slide. This is the way we have prepared. So now, so what are the challenges we are facing? The first thing, availability of the genuine raw materials. You may ask me, sir, you are already preparing your own, or you are already farming your own products, own raw materials. Then availability of the genuine raw materials, how will be a problem to you? So initially, before we approach it for the farmers, we have searched for the genuine raw materials. So we are fed up with the raw materials because simply we searched for more than 100 suppliers. No one is provided a genuine raw material for us. So as we are fed up with those services, next we went to the Botanical Survey of India. We have taken some samples from them, and we have given it to the farmers, and we are making them to work for us, and we are taking back the raw materials from those farmers, and we are giving them the payments in this way. So this is the way we have avoided this challenge. Next, purification. So whatever we have discussed here are getting of active fractions. So this is not a simple task. So purification of active fractions is required high technologies and separation techniques, and it also requires heavy equipment. Everything is needed. So this is also a challenge, but we can avoid, or we can answer this challenge with collaborations. So how can we answer this challenge with collaborations? Is one example what we have done, I can tell you. So there once after complete of Soxhlet extract, we try to move to the 50 kg batch. So in Osmania University, as we are working in Osmania University, we don't have that much capacity. So when we approach a technology business incubator, our CEO is Jaharullah sir. So he has a contact. He has a company that can do 50 kg capacity Soxhlet extraction. So he made us a link with that company, and we went to them. We reached them, and once we reached them, we asked them to, sir, this is the material you need to give us to the Soxhlet extraction with all these solvents. So kindly give me the price. So we have outsourced this thing because we don't have that much facility in our Osmania University. Because it is a university, it is only for R&D purpose. Now we are entering into the product development. So we need more and more amount of the extracts. So for that purpose, we are in tie-up with one company which is doing only extractions. So in this way, technology business incubation centers are helpful because they know where the required things will be done. How they because they are having contacts with them because they too supported them. So that's why in reverse they will support us. So this is the way. So that's why technology business incubators are collaborating units. So collaboration is the best thing in the startup ecosystem. Next, the third thing is also equipment for infrastructure and research and development. This is also a challenge. So, but here in this last challenge will be answered by the business incubation center. So they have given us the space. So initially we don't have any lab space, we don't have any equipment, we don't have anything, but what we have is only idea. So with this idea, we have made the technology incubation center people to analyze our idea. Once they have analyzed, they have accepted to our terms, and we have accepted to their terms, and they have given a space now in the Department of Botany, Osmania University. So they have provided a lab and facilities. Now we are doing the research. So, and we have developed the product now. So now we are enter into the manufacturing. So next. Next slide, please. Next slide, please. Next slide. So everything is fine. You have prepared the product, and everything you have prepared the product. It is very successful in R&D. Now coming to the marketing team. Always a business will stand only. Always a business will stand only. You have the profits. Only you have the profit.
Target market.
In order to show the profits, you need to analyze your market size. That means how many members will buy your compounds. So how many members will buy the things? So now, this one. So here I have, madam, keep the thing. Keep this. Thank you. Here. So how many your product? How many members can it use? That means these are the statistics from our India only. It is not entire world. It is India only. We have 41% of the obesity people, and in that one, 43% are suffering from heart disease, diabetes, cancer.
मुझे पता तो है, हर्बल में ये सब मुझे पता है.
Okay. Now, so all these are our market people. So those who are suffering from all these diseases, any one of the diseases can be our customer. So next one, next slide. Next slide. Madam, next slide, please.
हाँ चलेंगे. Next slide. नहीं, ये next slide.
Next slide, please. Next slide. Next one. Okay. Business model. So business model means, so how you are going to generate the revenue.
इसी में छोड़ दो, फिर धुल के रख देंगे अगर
Can you mute your audio, please?
देख के कहते हैं कि अब टाइम हो गया.
Somebody's
All
audio.
the delegates, please mute your mics. సార్ సురేష్ సార్ మీరు ఒక్కళ్ళు కొంచెం అన్మ్యూట్ చేయండి. అన్మ్యూట్ ఆల్ కొట్టాను.
Okay.
Yes sir.
Okay, madam. So can you go to the last slide, please? Business model.
Yes sir. Yes sir. Okay sir.
So now, generating the revenues. That means you have prepared a product. If no one is there to buy, then it is a waste of the thing, right? So first we need to analyze the target market. So what is the target market we are having? We are having a plenty of target market who are having obesity patients, heart disease patients, everything, because obesity is the root cause for every disease. Most of the diseases, lifestyle disorders are linked to obesity, directly or indirectly linked to obesity. So 41% of the Indian population, that means around, I can tell you, 50 crores. 50 crores of the people are our customers. So per day, if we sell only one herbal tablet to them, also 50 crore tablets we need to give them. So this is the market size. You understand? This is the market size for this drug. So for this product. So now, how you are going to generate? How you are going to sell these things to the people? So that comes under business model. So we have divided our business model into 3 parts. So first thing, give them the active fractions. First thing is develop and manufacture the active fractions and sell these things to some other companies who are already playing in the herbal industry. Okay. Now, the second thing is plant-based new drug discovery. This is our major area. Major thirst area for biogenic products is plant-based drug discovery. And third one is customized B2B research services. So why we have divided our company in these three business modules means simple. First, we don't have that much money because drug discovery process needs some hundreds of crores. We don't have that much money, but we can, what we are having, our strength, we can do the research and we can scale up the process. And another thing, we can create the products. We can create the innovative products. So for innovative products, there are always need. Innovative products are always in need. So prepare some active fractions or manufacture some active fractions and give these active fractions to a formulation company that is in need of the formulations. So we are selling now in the first area, development and manufacturing of the standardized herbal active extract, active fractions. We are thriving in this area. And the second area we are thriving is customized B2B research services. So our major portion of the revenue is coming from these two sectors. So as we have started in January 2022 till now, this 6 months, last 6 months, we have made our turnover around 18 lakhs. So 10 lakhs from the research customized B2B research service and 8 lakhs, around 8 lakhs from the standardized herbal active fractions. So but we are concentrating on the plant-based new drug discovery only majorly. So that it is in a long term. So for next 10 years, we will work on this plant-based new drug discovery. Plant-based new drug discovery. And now for our sustenance, for our day-to-day sustenance, we are utilizing these two areas. So we are created the active fractions. We are directly selling those active fractions to the another companies, and some other companies are needed the research service, not the products. So we are giving the research service to them, our services to them, and in making their formulations and in making their extractions more useful for the business generation. So next thing, next slide. So operational business model. In which areas you are marketing your product. So you know this slide. So purified active fractions, B2B means business to business. That means we are a business and we are selling it to another business. So these purified active fractions, initially we are selling to the business B2B sector. That means another companies who are willing to buy these things, and these are also marketable products. Directly we can market these things too, but in order to market these things, we need more and more amount of the investment. So this is an investment centric area. That's why investment centric area. So this investment centric area needs more amount of the investment. So we need to wait for this is called white labeling. We are doing this white labeling. So all our white labeling products are marketed in B2B sector. So B2B, all these B2B are white labeling. So we have prepared, but we can't utilize our name. But here in this case, you can generate more amount of the money than the B2C. Because you are already having a rich research and development expertise in this thing. So they will pay you how much they want, how much you can ask. So next slide, please. So till now we have prepared all these. They have developed the technology for these products. These are all our products. So now our major product is ashwagandha 35% with phenolides, and the second one Google 40% Google lipid. 40% Google lipid. So this is the major one, anti-obesity drug. So anti-obesity agent. Major research is going on the Google. So these are our some of our products. So we are also having another range of products. Somebody stopped it, please. Okay, next slide, please. So next, coming to our active fractions. So these are our active. First one is okay. So this is our Usmania University. So initially, to test us, our Usmania TBI asked us to prepare a hand sanitizer as we are suffering from COVID. So we have prepared a hand sanitizer according to WHO formula, but it is pure natural. We have prepared it with aloe vera juice. With aloe vera juice, it doesn't contain any synthetic chemicals. And the next product is GSZ. The code name one is GSZ. So this is ready, and now we are going for the preclinical studies for this one. And CTL3, it is a bone disorder active fraction. We are isolated from the natural source. One plant source we have isolated, and this one CTL3. For CTL3, around we are having a tie-up of 15 lakhs for the next 6 months. And third one is PCB. This is a depigmentation pure natural isolated compound. This is a utilization for the depigmentation, and one multinational company is approached us for the development of this PCB into a serum, a skin serum. So now we are preparing the skin serum, and we are in due diligence process. That means total from design to final development of the product. So due diligence process is going on. This due diligence process cost is around 80 lakhs for the next 3 years. So now we are today why we are becoming late means because of this product only. Our VCs are consulted, called us to a meeting on this PCB serum. So this is the thing, and these are our products, and all these products, 4 products are now in the commercialization stage, and the PCB is also already commercialized, and we are giving on the white labeling. So further developments are needed from that company, so our client company. So and for these 4 products, we are having the 4 clients. And next slide, please. So these are some of our companies, client companies. Customizer B2B research service. What we are giving the research service. So we have given to these 2 companies. Another 3 companies are there, but we didn't add those things in this one. Next slide, please. So market opportunity. So this is what it is. So by 2026, $411 billion is the market size of the natural products. We are not aiming into the entire natural products and nutraceuticals to be our company to be the solution. What we are aiming is only 1% of the entire market share. Only 1% of the entire market share. So that is around $40 million. $40 million in the next 5 years. So we want to grow our company into a $40 million company in the next 5 to 6 years. But our Usmania Technology Business Incubator already have a plans for our takeoff for our next for our next 10 years. So next 10 years we are going to work with close with the Usmania Technology Business Incubator, and we will make sure, and we are thinking that we will make sure that we will reach to the $40 million potential. So this is the thing, and kindly go for the next slide. So this is the thing, and this is one poem: the wood lovely, dark and deep, but I have promised to keep, and miles to go before I sleep, and miles to go before I sleep. This is from Robert Frost. So simply, the woods are lovely and dark means the coming days are dark and deep. because we don't know what is going to happen in the coming days. But I have promised to quit. Our promise is to become a $1 million company in the next 5 years. $1 million company in the next 5 years. That's why. So before, when I sleep, when our company will happily sleep, is only achieving after the $1 million company mark. That is our aim. That's why before I sleep, we will go through that thing. And thank you for the NIPS whole team, principal sir, and remaining member, honorable members. If you are having any doubts regarding technology business incubators and how they work and startup ecosystem, you can ask me.
Thank you, sir. Thank you, sir, for your wonderful session. So, just my personally, I have a small doubt, sir.
Tell me, madam.
So, what is the reason for you to drag for or to convert to startup, sir, from your teaching profession?
From teaching profession, it is not that thing, madam. Initially, I'm a person who always in touch with R&D, madam. I'm not a purely, I cannot say I'm a teacher. I'm very much inclined to the research and development departments. So initially, I worked in Dr. Reddy's. After my M.Pharma, I joined in Dr. Reddy's, and the working style is very toxic in Dr. Reddy's. So I left Dr. Reddy's and I joined in the institute college. So there I have done my research works, and there too I have I make the principals and everyone to move towards the research, not towards the education, because education it will be taken care, but research and development, it is a huge issue. So go for the and go for that way. Both the college management, they have accepted and they have given their consent to go for the R&D. So from their support only, I have published around 8 in international journals, and all are having very good impact factor. So after that, we thought that this type of the products are not there in the market because toxicity issues are there, but people are still using those products. So why can't we give a try to this thing? So like that, the idea came into our minds, and we started. And in between, the process went on, and everything is going on smoothly. But a long run, and it is a very hard work. It contains very hard work, because you won't believe us. No one will believe us that we have started our company with only 50,000 rupees. It's 50,000
Okay,
rupees.
sir. Sir, one more doubt, sir.
Tell me.
Actually, while in the process of your explanation, you had just given us, when you have an idea for a startup,
Okay.
you told Indian government is going to support by incubation something, sir. Can you just brief out in detail, sir?
Yes, madam. Oh, now, madam, so first we have this idea. So let me tell my story, this company story. We are having this idea. Me and Sudhir, myself, we are having this idea. Both are. We know that we have seen this problem. So we want to give a solution for this one, but we don't know whom to consult. But we know Sujata madam. She is because initially for our research work, she is the authenticator. She used to authenticate the plant material. So anyway, we are working on the plant material. So for every plant, we need to go for the authentication. So as she is the major one here in the Osman University who authenticate the plants. So we went to her, and one day we explained her the strategy. So this is the problem, this is the thing, and she was interested in our idea. She told us, only she told us, come to Technology Business Incubator. We are having incubation center. We will support you. So like that. And initially she just worked as a mentor for us. But after seeing our hard work, she started. She told us, I am going to join in your company. I'm going to join in your company, and let us form a company and take it into the next level. So this is the process. We haven't started to we haven't think to start a company and go for the research. We are in the research. So this research, it developed into a company.
Yes, sir, but many of them have ideas. As you
Yes.
said, maybe of a part of an academics or part of their student life, many of them
Yes.
are interested to do some innovation. But the
Yes.
main problem is they'll go some forward and at a certain stage, they don't know what is the step to forward.
Yes, yes, that
So
is
at
what
that time,
that is what you need a mentor. So
yes, sir.
we are doing the research and we told this idea to one person, and she knows the technology business incubation. So she knows the incubation process. So there in the once you enter into the incubation center, so what is going to happen means the company formation things are the idea of taking forward a small idea into practical form. It is taken care by the business incubators. It is taken care by the incubation centers. These incubation centers. These incubation centers also called Atal incubation centers.
Okay,
So
organized by IAC.
yeah, not only IAC, so many incubation centers are there in India. So literally there are 752 incubation centers in India entire.
Okay.
So here in Usmania University, one incubation center is there. JNTU Hyderabad is there. JNTU Kakinada and CCMB, IICT and IICB. So many things are there. Every national institute is a research center, incubation center now. So now in our story, we came with idea. Me and Sudhir came with idea and we consulted madam. She mentored our idea. She mentored our idea. When she is mentoring, we have only 50,000 in our hands.
Okay.
Only
So
50,000
was
in our
also
hands.
listed.
Yes, yes. And we have purchased some small chemicals and raw herbs, and we have given some small miniature type, small product, and we have tested it. Once the preclinical studies are came, then madam entered into our company. So we have formed a company. So once this has happened, the longer terms, the people where we are associated with some people. Now, what happened? You won't believe. The Usmania University is taking up a share in our company, and Usmania University itself invested in our company. So yes, now the 10% share of the company is for Usmania University. Now, because of the 10%, they are sharing everything. They are giving the permission to any lab. We can enter into any lab. We can do whatever the work we want.
It's
So this is
advantage.
the way. This is the advantage. So this is what incubation centers are doing. They will take up some of your share, and you should be ready to give the share. If you are not ready to give the share, then you will stop your idea. That is the
Sir,
thing.
as of your explanation, many of your works are also purely isolated herbal products, right?
Yes.
Yes. By using column chromatography, I hope
By
the
using-
isolation is there.
No. So the different techniques are there. Not only column chromatography. Column chromatography is a small part.
Okay.
So separation techniques are so many things. Briefly speaking, the major separation techniques are 6 to 7 types. So we can go any one of the separation techniques, but as it is very familiar with the people, pharmacy people, I told the separation techniques, but so many other separation techniques also there.
Industrial level, many.
Yes.
Okay. Sir, I have one question from Dr. Vijay Bhaskar, sir. So,
Okay.
sir asked properties and hexane identification test if present in fraction. Maybe sir is asking about the hexane, a solvent.
Sir, can you explain your question? I didn't get it.
Sir, Vijay Bhaskar, sir. So, just I'm asking you to unmute. You can even directly ask the question, sir. Sir, hope you are online. Vijay Bhaskar, sir. And Shushmita Manas, ma'am, you have raised your hand. So I have also given. Sir, Vijay Bhaskar, sir is online. Sir, Dr. Vijay Bhaskar, sir.
Yeah, madam.
Sir, you can
sir was
interact.
told, yeah. That sir was told active fraction based on the product. The hexane is having the active chemical contents. But what is the chemical test for needed for the hexane if it is present in the fractionation?
Sir,
What is the chemical test?
what is the chemical test? Generally,
Identification test for hexane solvent. If it is
hexane
present
solvent,
in your
so
extract or fraction.
Yeah, I understood your question. So generally, you will do the preliminary phytochemical analysis, right, with the different chemicals. Suppose Lieberman-Butcher test for steroids, glycosides, and ferrichlorate test for tannins. Remaining, so many things you will do. So in that thing, generally in the hexane, as hexane is a non-polar solvent, you will try to see the only non-polar chemical components. So like steroids, fatty acids, fats, and resins. So these type of the non-polar compounds only you will observe. So if that in that non-polar compounds, there may be 20, 30 compounds present, phytochemicals present in the hexane fraction. In that one, we need to separate the things by using different chemical separation techniques or column chromatography, whichever it is applicable or whichever it is economical. So based on that one, we'll do. So after chemical testing, so it may be steroid that is responsible for the action, or it may be a resin that is responsible for the action. We don't know what is that one. When we will know only after separation of individual component. That means simply isolation. That is a biggest. That is a bigger perspective. In the case of R&D, that means that comes under complete drug discovery, plant-based new drug discovery. We are not going into that one. What we are doing is we are into the thing where you are getting the activity. So take that fraction and do the chemical test and do the in vitro and in vivo assay and make sure that it doesn't contain any toxicity. If it is not toxic, then we will convert it into a formulation. If it is toxic, it is active. Listen this carefully. It is active. That means it is having anti-obesity or anti-diabetic activity, but it is toxic. We should not use that fraction. So now that's why we need to purify that fraction. So which component is giving the toxicity? We will eliminate that thing from the fraction and we will make that into active fraction and we will make it into a product. Did you get my point, sir?
Okay. Basically, that hexane solvent is a cleaning agent in industrially, but in after fractionation, if it is having the hexane, how you identified if it is present or not in hexane in your fractionation or extract?
For each batch, we will go for the GCMS analysis.
CC?
GCMS analysis, gas chromatography, and coupled with mass spectrometry. We will identify with the GCMS analysis. Once it is present in the final compound, hexane is not useful. It is a toxic. So if it is not present, it is there in the more than the limits. What we have to do is we have to purify it again. So for that purification, there are so many techniques are there. We will those industrial techniques we will utilize and we will go for the thing.
Okay, one thing is basically in previous so many years, all are using the non-polar to polar solvents. But why don't you go to Zeta fractionation?
Zeta?
Do you know idea about the Zeta fractionation?
No, sir, I didn't have any Zeta fractionation.
Basically, in phytoconstituents, we are using the non-polar to polar, but the one mechanism is there. But we are without using the solvent, that is the Zeta potential. We have to try once in our university.
Okay, sir. But Zeta fractionation, you are telling that it is maybe a R&D. It is in the R&D stages only. But for manufacturing scales, think about it in the manufacturing scale, because your product should reach thousands of people. In order to reach thousands of people, you need to do it in a kgs manner. Whether this Zeta potential based fractionation is applicable for that much amount of the components or that much amount of the raw materials, it is useful. If it is not useful, then we will go for the remaining. We need to go for the alternative method. So first we need to check whether the method is applicable for the larger amounts of the components. So
okay, which solvent is suitable for the isolation of the bioactive compound?
Sir?
Which solvent is suitable for isolation of the bioactive compounds?
That is based on the trial and error method, sir. You can't say that one. You have to utilize this. You have to utilize different solvent systems.
Basically, the hexane is especially useful for the isolation of the oil compounds, just like volatile oils.
Okay.
But you are using hexane, your solvents are, you are telling no, ethyl acetate, hexane, CCl4.
Yes.
Which is the active chemical constituents separation of bioactive and plant constituents, which
Yes,
suitable solvent?
that's what I'm telling, sir. Each one depends on the trial and error basis. So in which solvent it is going to come, we don't know initially. Are you getting my point, sir?
Yeah.
Yeah, initially we don't know from which solvent it is coming. Once first we do the extraction. After doing the extraction, we will go for the isolation based on the activity. If it is the extract is active, then only we will go for the fractionation. After that fractionation, we will go for the isolation of the component. So here that we can't tell directly. No one will tell directly that if you utilize this solvent, it is going to come. After doing some trial and error methods with different solvent systems and different pHs, not only solvent system. So don't think that only solvent systems will work in the natural products industry. That is not the case. We need to utilize different buffers. We need to utilize different pH solvents. So many things are there. Though we need to utilize all those things. After that only we'll come to one conclusion. Then we will write it in a procedure manner. So this procedure is applicable for this one.
Okay, I will contact in future.
Okay, sir.
So plenty of doubt is there in natural products. Basically, you told before that herbal medicines are already saturated.
Yes.
But why don't you come? You don't go to mineral sea sources, marine sources.
Identification is a problem. Identification is the biggest problem.
That is the advantage. In China, Japan also, they are forward to 50 years advanced technology. But India also, now we are isolating this method, sir.
The marine, sir, the
funding also in university.
Yes. Let me tell you one thing here in the case of marine sources. So first thing, you suppose you became successful in isolating one very good compound. Suppose antihypertensive drug you have isolated from the marine source. Now the problem with the marine source is harvesting. Did you get my point? So bulk harvesting is not possible in the marine sources. So where you will harvest those things? In sea only, right? So how you harvest in the sea? It is a very big task. And so once it happens, you are harvested also. But if yield is less, then the product value is more. Yes or no?
Yes.
So already in the herbal market, you are having ephedrine. So which one you will choose? Ephedrine comes for 40 rupees, and your drug is coming from 40,000 rupees, as it is a marine source, but it is potent. I can tell you, it is safe and potent. But ephedrine is not safe and not now it is relatively safe and potent. So which one you will incline? Surely for ephedrine, not for your compound, right? So we need to check. In order to you want to make it successful as a product in the market, you need to go according to the market. Did you get my point, sir?
Yeah, according to the market, but the research is forward and advanced means that's the better one.
Yes, sir. Yes, sir. Whatever you said, it is applicable for R&D scales. But from R&D scale to manufacturing scale, so many issues will come. And once it made into the manufacturing, also the costing also comes into the picture. So I can tell you, there are so many natural products are there that are most active than the synthetic drugs, but they are not marketable because of the high prices, non-availability, and adulteration. So that is what we want to cure. Those are the issues, and we want to solve those issues.
Okay.
But sir, from plants also, I hope we need more amount of crude drug, no sir, for isolation or for extract also. From herbal extracts also, we need to get more.
Yes. So now tell me, I can tell you one thing. So before before 2010, the total ashwagandha production from Telangana and Andhra Pradesh is 100 hectares.
Okay.
Now it is 80,000 hectares.
Okay. So production has been.
We need to motivate the farmers to go for alternative. So medicinal plant cultivation is the biggest market in the next 10 years. And active fractions based market is also the biggest market in next 10 years because, and thanks to COVID.
Because most of the thing has been dragged into the scientific.
And no
The
synthetic
eye is open to science now.
no synthetic medicine cured that one. That is the thing. So is there any other questions?
Sushmita Manas ma'am, just you have raised your hand. If you want anything, interact with this, sir, please. So Dr. Vijay Bhaskar, sir, hope your doubt has been clarified, sir. Sir, as a token of our gratitude, so we want to present a small certificate, sir. So please
Okay.
expect that the certificate has been given
Thank you,
to all.
madam.
Now that will forward
Okay,
you.
thank you very much. Thank you very much
sir.
for the management of the NIPS and principal and especially you, madam.
Thank you so much, sir, for your acceptance and sparing time. Though in spite of delay, we have managed it in a cool way. So the only thing, because some
Yeah.
network issues initially, there may be some voice disturbances, but everything went on good, sir, later. Thank you
Okay.
so much, sir. So in future, in our
Thank you,
students also, if they want any extraction or if they want further, give priority
You
importance for our
can
students.
you can contact us, madam, at any time for R&D and for manufacturing help. We will help because I know the difficulties and pains in this sector. So I can help with open hand. You can come to our doors always open for you at for NIPS.
Sure, sir. Thank you so much, sir.
Okay. Thank you, madam.
Thank you, sir. So delegates, the link for your certificate or feedback will be given in the afternoon session. By 2 o'clock, the second session will be started on IPR. So I request all the delegates to be back by 2 p.m. to join another awareness program on patents, IPR. So thank you one and all for your kind patience and listening because of my delay. So, and some network errors also. So I request all the delegates to again rejoin the session with the same link by 2 p.m. Thank you. Thank you one and all.
Thank you, ma'am.